look at the central nervous system, I'll put the pointer on here, just to remind you that the central nervous system is the brain and the spinal cord. The peripheral nervous system, as you remember, is everything outside of the brain and the spinal cord, and that's the nerves. We're going to look quite a bit at the peripheral nerves in lab tomorrow. And then we have the ganglion. We have different types of ganglion, and we'll look at that. Those are the major parts of the peripheral nervous system. The central nervous system is composed of different areas. Rather loosely, we differentiate the central nervous system into the white matter and the gray matter. When we say the white matter and the gray matter, this is kind of what they look like under gross appearance. In other words, the unaided eye. It's not what it looks like under the microscope, but it's what it looks like without the microscope. The white matter is composed primarily of axons, most typically, myelinated axons. So this is where we're having transportation of information throughout the white matter. The gray matter is more of your integration region. That's composed of your cell bodies and your dendrites, maybe a little bit of unmyelinated axons. The gray matter has another term that we'll use more than once today, and it's called neural pile. The neural pile is equivalent to the gray matter. In the brain, the bulk of the gray matter is to the periphery of the brain, and the bulk of the white matter is more to the deeper structures. So surface is gray matter, and deep is white matter. In the spinal cord, it's the opposite. The gray matter is the deeper layer, and it forms the H, or butterfly shape, deep into the spinal cord. And the outer areas is the white matter. So they're opposite in that way. We do have, in the brain, patches of gray matter within the white matter, within the deeper white matter. And that's usually called ganglion or nuclei. And so patches of gray matter deep in the brain are nuclei. So this is telling us all of these kind of things here. These next few slides are basically to give us an orientation of the different areas of the brain. And again, we'll be focusing on the brain and the spinal cord, I should say. We're going to focus on primarily three of these histologically. When we say caudal, caudal means tail. So if we're going from caudal, that's sort of where the brainstem is. Rostral means nose. So from caudal to rostral, from the developing central nervous system, we have the spinal cord, the medulla oblongata, the pons, the cerebellum, the midbrain, the diencephalon, and the teleoencephalon, which is the cerebrum. We need to protect the brain structure, so we have the scalp, we have the skull bones, and then we have the three meninges. The three meninges are membranes surround both the brain and the spinal cord. They're protective in nature. They hold the structures together and also protective. The most superficial meninge, you may remember, is the neuromotor. Then the middle meninge is the arachnoid. It gets its name because it's spider-webby shape, arachnoid spider. Okay? And then finally, the deepest layer, which is attached right to either the brain or the spinal cord, is the pia mater. The dura is the tough mother. The pia mater is the soft or delicate mother. You may remember from some other source. So we have those three layers. When we look at the brain is thrown into ridges or folds called gyri. Gyri is plural, gyrus is singular. And the depressions between the valleys, if you will, are called sulci, or sulcus is singular. Here in this photograph, the pia mater and the arachnoid is still intact along with the blood vessels. The dura mater is cut, it's right over here on the edge, but it's been cut there. Here's the dura mater. Maybe I'll close this door since they're having so much fun out there, but it's gonna make it pretty dark, but maybe that'll be all right.
Promise not to fall asleep.
I wouldn't know if you did, Ron. This is looking at the brain from the upper part, and this is the cerebrum. The cerebrum makes 80% of the brain. So 80% of the brain structure is cerebrum. We have a longitudinal fissure that incompletely separates it into right and left hemispheres. This is cutting all the way through the brain. We're not wanting you or necessitating you to memorize all these layers, but just to give you an idea, the cerebrum is all of this part here. If you can see the little red laser dot. And then here's the cerebellum. And the spinal cord would be attached down here to the medulla oblongata. So we're going to be looking histologically primarily at the cerebrum and the cerebellum and the spinal cord. And we won't really be paying much attention to the other areas here. The brain has some ventricles, open spaces right behind this laser pointer is on both sides is the lateral ventricle. The lateral ventricle goes into the third ventricle, which goes down this cerebral aqueduct to the fourth ventricle, and then it goes down around the spinal cord in the central canal of the spinal cord. The reason we mention that is these structures are filled with cerebrospinal fluid, and they're made by structures called the choroid plexi. Plexus is singular. And so we'll be seeing some of those structures. And they're lined, all these ventricles and cerebral central canal and all that, by ependymal cells, one of the glial cells that we've mentioned. This shows you that we can also divide the cerebrum into lobes. This shows you other areas of the brain, like the diencephalon and the midbrain and the pons. There's the cerebellum, the medulla oblongata. Cutting the brain at a rather odd angle, we see the white matter is deeper, so we're sort of tracing the white matter here, and the gray matter is more superficial. The gray matter there in the cerebrum can also be called the cerebral cortex, and we do most of our cognitive activity in that cerebral cortex. So that's the gray matter, and you can see the photograph of the actual brain is showing, it looks more grayish, and the gray matter is white in the deeper areas. When we look at it histologically, the white matter, which is, if we're tracing from about here down, it's over to our left, all this is white matter. These are axons. This is a special stain called osmium stain. It stains the myelin. See the dark purple rings? That's myelin. So all these are axons with surrounded by myelin. We've got a lot of myelinated axons here, in cross-section. Over here is the neural pile, that's the gray matter. So gray matter here and white matter over here. This is a different stain. This is showing the gray matter, and these largest cells are the neurons. There's a neuron. You can see the nucleus. We're sort of tracing it around here. And there's the nucleolus. Here's another neuron with the nucleus and the nucleolus. There's another one. So these largest of cells are the neurons. These smaller nucleated cells are glial cells. We have different glial cells, you may remember, depending if we're in the central nervous system or in the peripheral nervous system. In the central nervous system, the oligodendrocytes and the astrocytes are the most common. The astrocytes are the most abundant of all the oligodendrocytes. In addition to the oligodendrocytes and the astrocytes, we have microglial cells, which are phagocytic, part of the immune system. And we have the ependymal cells, which line the ventricles and the central canal, where the cerebral spinal fluid is located. In the peripheral nervous system, you may remember that we have Schwann cells, and we have the cells in the ganglia, the support cells, which are often called capsular cells or ganglionic cells. So we'll be looking at different cells here. Notice that the glial cells are quite a bit smaller than the neurons typically. This is from your book, but here we see some oligodendrocytes and astrocytes. This image here is magnified more over here. And what we're seeing is these small rounded nucleated cells are called the oligodendrocytes. They often, not always, have a little bit of white space around them. The astrocytes tend to have a little bit more elongated nucleus and a little bit bigger than the oligodendrocytes. These differences are rather subtle. If you use special staining methods, you could differentiate them in much greater degree of specificity. For instance, the astrocytes have more of a starburst shape if you see all their processes. But here we're only seeing the nucleus. This is another image. These slides here where you have comparisons, make good questions on exams or tests, because we can differentiate. These are oligodendrocytes, a little smaller, often having a little white space in them, whereas the astrocytes are a little bigger, typically in a little more elongated nucleus that we're seeing there. And the neurons, of course, are considerably larger. So these are the neurons, and then these are the oligodendrocytes that we see, and these are the astrocytes. Tomorrow in lab, we will try to differentiate between the neurons, the oligodendrocytes, and the astrocytes, but we won't try to differentiate. Well, the ependymal cells are easy to find, as we just look in the central canal or along the ventricles. But the microglial cell, they're the smallest, and they're really difficult to differentiate from the oligodendrocytes. So we won't really try to do that on the slides. This shows you a particular stain, Cresol Violet Luxol Fast Blue stain, long name. In this case, it does make things perhaps a little bit more distinctive. These larger cells are the neurons. So we see a number of neurons there, the largest. Then the more intermediate ones that we have the yellow arrow on a couple of those, these are the astrocytes. And then the smallest, see the nuclei here, see the little white space around it? That doesn't always happen, but if it does, you get a little bit more confident it's an oligodendrocyte. So here's an oligodendrocyte, and here's some, see the little white space around there too? So these are oligodendrocytes, and these, at this magnification, we're comparing the oligodendrocytes versus the astrocytes versus the neuronal cells. What is all this blue stuff here? These are like the dendrites, the processes.
Trinity has a question.
Oh yeah, go ahead, Trinity.
Are we not seeing the nuclei of the astrocytes, or how come they're not more elongated?
Yeah, that's a good question. We don't see the complete, if you see it, do you see the red pointer razor? Do you see how it is a little elongated? And this is, we have the nucleolus, and it's a little elongated, but yes, it's not as distinctive as back here as we've gone to a little bit larger size. So if we blow this up, the elongation is not as enhanced as when it's bigger like this. And so, you're correct that doesn't look very elongated. Here, we would be going mostly by size differentiation. Back here, we are differentiating by size, but we are also somewhat differentiating by elongation of the nucleus. We could overemphasize that elongation. Let me show you. This is an astrocyte here, based on size, and its nuclei doesn't look elongated. So you're correct that is somewhat of an incomplete differentiation. So we're looking somewhat for trends, and the trends here are maybe a little elongated, but they do look more rounded here, don't they? The nuclei. Yeah, sorry, that's not a huge, well, maybe it's enough help to differentiate them. When we look at them under the microscope, we'll have a bit of a challenge, perhaps differentiating them, the two. These are the microglial cells, and you can see they're quite small. This, they used an immunohistochemical mechanism, and that's how we really pick them out. You're picking out the whole cell here. You might say, well, these look bigger than some of these others, but that's because you're using this technique. You're seeing the whole cell, not just the nucleus. So we won't really be able to differentiate these from the oligodendrocytes, for instance, on our slide, from our slide box. This is a different type of stain. Look at the neurons here, very nicely laid. This is the pale nucleus, and here's the intense nucleolus. And you can see the nucleus and the nucleolus. Sometimes you can see some processes. It sort of depends on the angle that you caught it at. These are myelinated axons here. Axon, myelinated. This is one that, yeah, go ahead.
You go back one more. So on C, the kind of darker ones, those are microglia. One more over. The one on, yeah, that one.
Yeah.
Those are microglia?
Yeah, they're just at a lower magnification than this one.
Okay.
They look like this in the diagram,
Mm-hmm.
and they kind of look like that.
Yeah.
But again, based on special staining, we would have to, if we were just shown this, we'd probably have to say, what were you staining for? Microglial cells or oligodendrocyte? Because oligodendrocytes have few, their name means few short processes. Oligo means few, short processes essentially. So
Yeah.
that would be difficult to differentiate.
And those are only found in the central nervous system?
That's right.
Okay.
Microglial cells, astrocytes, oligodendrocytes, and ependymal cells are only in the central nervous system. Schwann cells and these capsular or ganglion support cells, they're only in the peripheral nervous system.
What about satellite cells?
Satellite's the same thing as capsular or ganglionic support cells. Yeah, satellite, meaning they go around the neurons like a satellite goes around the earth. Capsular means they're forming a capsule around the neurons. So a couple of alternate names, but they're all the same cell.
Okay. Thank you.
This is the same one we looked at with gray matter over here and gray matter over here. When we look at the cerebrum, even though it's 80% of the brain, we're not going to spend a huge amount of time on it. Because histologically, going from superficial to deeper, it has 6 layers, with the molecular layer being the outer layer, and then these 6 layers here. But these layers under H&E staining, which our microscope slide box has, are extremely difficult to differentiate. And we're not going to ask you to differentiate it from our slide box. Just to show you this point, this one to our left is a methylene blue, and this is an H&E to our right. This shows you that we're taking it from the cerebrum. Now, when we look at this, we can see the molecular layer on our H&E, and that's fine. And we can see that there may be layers, but unless we have some help over here, it's pretty hard to differentiate these layers. Can we really see 6 layers if all we had to look at was the one on the right? So we won't be asking you to identify these layers on the microscope slides, except we want you to identify it as the cerebrum. So we want you to be able to identify this as the cerebrum, even under our microscope slides. Well, what about under these digital images? Well, if we have it stained like this, then yes, you can differentiate these layers. If we showed you one with these layers sort of mapped out for you and said, what layer is this? Then we would ask you to know that. On the other hand, over here, we would say, no, this is the cerebrum, no, this outer, more superficial layer is the molecular layer. Other than that, we wouldn't expect you to be able to differentiate them out. We'll be coming back to this, but the pia mater is right here on the surface, from about here to here. It has some blood vessels in there, but this is the pia mater on both sides. The pia mater attaches right to the nervous system's tissue, the brain, the cerebrum in this case. We'll see some more images of similar to this, but this is the pia mater here. Do you feel okay of what we're asking you on the cerebrum, how to identify it? If it's on a digital image and it has a specialized stain with the layers marked, then we might ask you to know these layers, which are shown here, by the way, the previous slide. If it's an H&E staining or such as under a microscope, we would ask that you know that this is the cerebrum, and maybe this is the molecular layer, maybe, you know, the pia mater, but we wouldn't ask you to know the different layers or be able to separate them on that basis. Our next is the cerebellum. The cerebellum, we will ask you to know the layers because they're quite easy to differentiate. The gray matter, the cerebellar cortex, is subdivided into the molecular layer, the Purkinje cell layer and the granular cell layer. Deep to that is the white matter. We'll want you to know all of these 4 layers, but they're quite easy to differentiate. This is part of the cerebrum up here, so let's not worry about that. What we're looking at down here is the cerebellum. This is what it looks like. Looking at the cerebellum, this white matter pattern that you see here, that's called the arbor vitae. That is the tree of life, because it looks a little bit like a tree. When we look at it histologically here, we see these layers. This outer one here is the molecular layer that I'm sort of tracing around here. Then, at this magnification, we can't really see it very well, but the layer right here would be the Purkinje cell layer. Then we'd have the granular layer, that darker, a lot of nuclei in there. And then this little strand right here is the white matter. So those are the different major layers. What is this right here? This is the choroid plexus. This helps produce the cerebral spinal fluid. What is this here? That's the pia mater. It's a little pulled away as an artifact. In life, it's right snug like this shows, but here you can see it being pulled away a little bit. So all of this coming around is the pia mater. There's some over here on the cerebrum as well, pia mater. Now we'll go to a higher magnification. So we're seeing this, and now we're blowing it up. Here's our outer molecular layer. This is pia mater right here. Coming here. Part of it may be torn off. And then we have the Purkinje cell layer here. You see these big cells right there and there, and that's the Purkinje cell. So we call that the Purkinje cell layer. We'll blow that up even bigger in a minute. Then we have the granular cell layer. All that's the cerebral cortex, or cerebellar cortex. And then we have the white matter, which is the deepest.
Is the Purkinje cell layer like 1 cell thick?
Yes, 1 cell thick. I mean, you might have some support cells, but the big neuronal cells are called the Purkinje cells, and that's 1 cell layer. See how we might have little small ones around them, but essentially the big neuronal cells are 1 cell thick. That's right. Now we've really blown it up. We've taken this layer. These are our Purkinje cells. You can see the nucleus and the nucleolus and some of the processes. Here's our molecular layer. That's that lighter stained area. This one with all the nuclei is our granular cell layer. And this in between them is our Purkinje cell layer.
Are each of those like tiny, like dark purple dots in the granular layer? Are those each cells, or?
Those are each cells. And some of these are neuronal cells, but a lot of these are glial cells.
Something.
Yes. And one of the reasons they call it the nuclear cell layer or the granular cell layer, you can see that we have those names, is because you just have all of these highly visible nuclei. Notice the molecular layer has a lower abundance of cells by quite a bit than the granular cell or nuclear layer.
Within regions of the brain, like the cerebellum and cerebrum, is there connective tissue, or is it pure, like, just neuronal?
There is connective tissue. Essentially, the meninges are connective tissue, for instance. But primarily, the connective tissue is holding things together. It's not running through the tissue, by and large. It's not like the peripheral nerves where you have connective tissue layers around every sublayer. So it's more holding it all together, but not really interspersed amongst it. This is the Golgi staining technique, which stains some of these. And you can now see these Purkinje cells with their processes and their nuclei. So this is kind of what they look like. Here, you can sort of see it, but here you can see the branching much more easily. This is also, this is somebody from, Cajal was a very famous histologist, and he drew this in Madrid in 1952 using the Golgi stain. And so he was one of the first ones to sort of elucidate these kind of Purkinje cells. You can see he's a better artist than I am. Makes pretty precise drawings there. The meninges we've mentioned. The superficial is the dura mater, and then the middle is the arachnoid mater, the arachnoid, and then innermost highly vascular pia mater. The brain is highly vascular. Of course, it needs a lot of oxygen and nutrients. It uses glucose primarily for its as a nutrient source. And then, of course, it needs oxygen. The choroid plexi, or plexus is singular, produces cerebral spinal fluid, and the glial cells, the ependymal cells, line all the ventricles and the central canal. We'll see those in lab pretty clearly. Cerebral spinal fluid circulates through this and eventually comes to the surface of the brain and the top of the brain and gets reabsorbed into a structure, a venous structure called the superior sagittal sinus. If this reabsorption is blocked in utero, in the uterus, that fluid backs up and compresses the brain tissue, and hydrocephalus can occur, which can be quite devastating.
I have a question. What does it mean by infrequent desquamated cells?
So the cells lining the compartments there can sometimes slough off into the cerebral spinal fluid. And so, although the bulk is water and ion, a little protein, not near as much as the blood plasma, a few white blood cells that sneak through, but not very many. And we get a little bit of cells that have sloughed off into it. We generally don't find red blood cells in there as well. When we look at the meninges, the outer superficial layer, the dura mater, then the arachnoid, those are generally in life pretty much in contact with each other. Then we have a subarachnoid space, here on the diagram, then we have the pia mater. So there's a space beneath the arachnoid, deeper if you will. We'll see that sometimes when we make a slide, the arachnoid will artifactually be pulled away from the dura mater. That's an artifact, not a reality in vivo, in life. This is the dura mater we're seeing here. Here we see it maybe a little bit better. Here's the dura, and here's the arachnoid. Here's the subarachnoid space, and here's the pia mater, and here's the actual brain tissue or spinal cord tissue. We have blood vessels going through the subarachnoid space and some of the pia mater. Now in this view, we have an artifact. Here's the dura. This arachnoid spiderwebby shaped thing in life would be attached up here. There's no space. So this subdural space is an artifact. Sometimes you've heard perhaps of a subdermal hematoma, and you can have some bleeding into this area that makes it not only filled with blood, but it compresses the brain and can damage the brain, kill the person potentially. Sometimes they have to drill down through the skull and relieve that pressure because it has really nowhere to go, not much of a place to go. Some of it may leak out with the cerebral spinal fluid, but if it's leaking too much, it's really gonna cause a major problem. So the dura, mater, and then the arachnoid, which normally would be up attached here, then the subarachnoid space, which is normal, not an artifact, and then the pia mater here with some blood vessels. And then the tissue, the brain or the spinal cord. Here's again a diagram from a book showing it. And here's the dura and the arachnoid, again being somewhat pulled away, and then the pia mater down here. We produce cerebral spinal fluid from the choroid plexi, and the cerebral spinal fluid circulates from the lateral ventricles, the third ventricle, down the cerebral aqueduct to the fourth ventricle, and it goes down around the spinal cord and the central canal. It eventually comes back up to the surface of the brain, above the brain, and drains into the venous system. And so you keep making it and then draining it. This shows you some of that area of circulation. Here's the choroid plexus that we looked at briefly before. Here it is at a higher magnification. The choroid plexus is essentially a modified capillary system. These are the ependymal cells lining all along here, these ventricles or spaces where the cerebral spinal fluid is circulating. Here's the ependymal cells. This would be the ventricular space or the central canal. It's the spinal cord space. And this is the choroid plexi, the choroid plexus. So it's producing the cerebral spinal fluid and circulating around. Just a bit more. This is the spinal cord. This has a special stain, as you can tell. This shows us the now deeper gray matter opposite to the brain. This is the butterfly or the H shape. And we're going to see some really big neurons there. We've already looked at it briefly. The very first lab we had when we were looking at the big motor neurons in the spinal cord, we were looking here. We also have the white matter around here. This is where the spinal nerves come in. And we can see the dorsal root ganglion. We'll probably see, probably on Wednesday when we look at the spinal cord. On some of them we'll be able to see the dorsal root ganglion, which is outside of the spinal cord. But we'll be able to see some of that. Now here's a stain. This is the dorsal root back of the gray matter. And these are neuronal cells. See how big they are. Notice the nucleus is paler and the nucleolus is darker. And then we have these little fibers running around. At this magnification, these little purple dots, those are primarily the glial cells, primarily astrocytes and oligodendrocytes. We can't really differentiate them at this magnification. I mean, we'd pretty much be guessing.
What's that big dark red spot?
This,
Yeah.
that's just an artifact. It either is a cell that got smushed during processing or some stain that just inadvertently got caught up there.
Is that the same thing with that, yeah, that?
This, I believe, is a neuron. It's just, we did get a little bit more intense staining of the nucleus and nucleolus, I think, in this orientation. So this certainly looks less typical than others. This one stained a little more intensely, but that one really picked up the stain for some reason. Here we see a little more, we picked up some stain there.
And those lines going into the white matter, are those the axons?
Yeah.
Okay.
And do you see this here? You have these are axons. The myelin is not showing here in this stain. So the sort of darker part is the axon, and the lighter part is the myelin in cross section. Now, if we stain with osmium stain, it would really highlight the myelin, and the axon would look white, like it's not even there. The W here stands for white matter, by the way, and the G for gray matter. So that's basically what we're seeing there. We'll kill your eyeballs here. So in lab tomorrow, we'll be focusing primarily on the peripheral nervous system. I think we do get into the glial cells, but we'll be looking at the peripheral nerves, and we'll be looking at the ganglion cells, ganglion outside, the autonomic ganglion, parasympathetic, and all that. So we'll be looking at some of that. And then eventually on Wednesday, we'll look at all these things like the spinal cord and the cerebrum and the cerebellum and all of that. And so that's kind of where we're headed. So then, just to remind us, the Wednesday morning, we'll be looking at the cardiovascular. That one's kind of fun. We'll look at a bunch of blood vessels and all that, and we'll start in the heart, and we'll differentiate different types of blood vessels. And because we see blood vessels all over the place, that's helpful when we start to see that. Wednesday at 2